Hi, everybody. It's Kim Curry here, bringing you the next steps of the My Gene project. Today, we're going to start back at the gene database at the NCBI website, same place we were before. And we're going to start off by searching our gene of interest. And I'm going to go back to my night blindness example, in which I found the gene GRM6 to have a connection to. So searching for my GRM6, I can see the records that show up and I look for the one that I'm interested in, the one for human here. And I'm going to open this up in a new tab. I do like to open up each page in a new tab so that I can easily go back to the previous page if I need to. So that brings us to this full report. We've been here before. You came here to determine whether or not the gene you've selected is protein coding. And you can see here all of the details are confirmed. It's from humans, it shows the lineage, it shows the alternative names for this gene, and then there's a summary of information, of course, included here of what is known about this gene and its translated protein sequence. Other details that we found last week were the location of this gene on the chromosome. And this week, we're going to expand a little bit further. And instead of looking at just the gene sequence on the chromosome, we're actually going to look at what the mRNA transcript or transcripts, if you have selected a gene that happens to have multiple splice variants. So looking here, I can see that there's an exon count of 11. There's 11 exons in the GRM6 gene. Not all sequences will list the exon count here, so you may have to look further into that, and I'll show you how to do that. And then the other thing we're going to be looking for this week is what direction, what's the orientation of the gene. So that is the orientation that it is read in when the mRNA transcript is synthesized. So in the case of GRM6, it is from right to left. And then scrolling down a little further into this graphical view, you may see a single, so I'm just looking in the genes area of this graphical view, you may see a single green line, or you may see multiple green lines. If you see multiple green lines, it means that likely this gene that you've selected has multiple variants. So that means that the pre-mRNA sequence is alternatively spliced into different final mRNA transcripts. So in the case of GRM6, I only see one green line here. And what you may notice is that there's large, thick boxes of green, and then there's thin lines in between them. And what that is the difference between the intron and the exons. So the exons, which get translated into amino acid sequence, they are shown as a nice big box. And then the thin lines here are the intron sequences. So if I click on that green line, it will actually expand and it will show me also a purple line and a reddy orange line. So what you're looking at here, and just a little bit about the legend of the graphical views so that you can follow along. The green usually is the sequence that's on the chromosome. That's the gene sequence. The purple line is the mRNA, that's the transcript that is produced from that gene sequence. And then the reddish line is the protein sequence. What you may have also noticed is that there's different coding systems. So all the records come with a letter and a number system shown. So if your letter starts with an N, that means that record has been experimentally verified. A scientist has looked at this and it has also likely been published as well. Sometimes the numbers start with the letter X, and in that case, those are predicted sequences. likely based on comparison to other already known sequences from maybe possibly other organisms even, that then those sequences are predicted to be the same as the other ones, but they're not confirmed. And then the second letter of the code here indicates what that record is of. So in the case of the letter C, that's the chromosomal record. That's coming from the chromosome sequence, that's the DNA sequence, gene sequence. And then the second letter may begin also with the letter M. So where's an example of that is this one right here. This one is an NM, and that M stands for the mRNA sequence, the messenger RNA sequence. And then this one, again, as an example, NP, the P stands for the protein sequence. So there's an easy way to check based on the code what those numbers actually, or what those letters as part of that record actually mean. So if you get lost at some point with all of the data you're collecting, then go back to that code and that might help you. So if I hover over, you've seen already, if I hover over one of these lines, there is an information box that pops up. So I'm going to hover over the purple line here, and this is the mRNA records. This is the record that we're interested in for this week. And if you go down, there should be a graphical view. So I'm going to open up that graphical view right now. Now, I will just add that if your gene has multiple variants, what I'd like you to do is to select the longest variant option, which you'll be able to see in the graphical view, which one is the longest, and then choose one other. And then you'll follow these steps for both of those variants going forward. So going to that mRNA graphical view, so this is the human GRM6 mRNA sequence. And having a look here, the color coding is slightly different. So I just want to look and make sure, okay, the length is 6,000 nucleotides. And if I go back to that full record, if I look here at the gene sequence, its length, full length is around 17,000 nucleotides. And then the mRNA sequence here, that only includes the exons, has a length of 6,000 nucleotides. Okay, so I know I'm looking at the right thing here in the graphical view. And what I can see is the breakdown of all of the exons for this particular gene. We know there's 11 exons in GRM6, and we can count them out here, and it's shown really nicely here as well. So this is the focus for this week. This is what we're interested in, is this exon breakdown if it applies to your gene. If your gene does not have multiple exons, you're going to need to look into why that might be. Is that true for all genes in the organism that you've selected, or does that just happen to be for the gene that you selected? Not all organisms have introns and exons. The other information that's sort of in the core here of this graphical view is related to the actual sequence details, and it can be quite interesting as well. For example, they know that this region of the sequence is likely a transmembrane region. So the protein, once it's been synthesized, has a transmembrane region, so it must go through the membrane. As you do more and more research for your MyGene project, you'll probably discover these details as well. So hang on to this graphical view. I think you're going to find it useful, probably for your presentation, but maybe for other work this term. And you can do that by selecting Download Printer-Friendly PDF version. There are two other records that I want you to hang on to for your mRNA sequence this week. One is the Genbank record, and the other is the Fasta record. So first, let's look at the Genbank record. This record is a detailed record of the mRNA sequence. So you can confirm the details here along the top to make sure you're still looking at the right one, and indeed we are. And what you'll see here also are some published articles related to this sequence. And in this case, there's quite a few articles. And if you click on any of these PubMed links, it will take you to those journal articles. And if you're logged into the VPN at UVic, there's a high likelihood that you'll get free access to the journal articles. So scrolling past all of these references here, you should come to an area called features. And this is where the features get broken down in a written format. So you'll see that the full length is 6,000 nucleotides for GRM6. That's what we found in the previous records. And here the first exon is from position 1 to position 45. And the second exon shown here is position 46 to 565. You may also find additional details in this record that are related to the protein. In this case, this is the coding sequence here. These are the amino acid sequences shown. And then there are some additional protein details in this record that you can ignore for now. We'll come back to them later. I'll continue to scroll down until I see my next exon sequence so we can see for all of the remaining exons, all of their positions and where they start and stop in the sequence. And then of course, at the very bottom of this record, you can see the entire sequence here. Now, if we scroll up to the top here, I was mentioning we also want to keep the Fasta record for this mRNA sequence. So let's have a quick look at that Fasta record. The Fasta format, for those of you that aren't familiar, is a very basic format that is recognized by a lot of software programs that analyze DNA sequences. So we're going to need this very basic sequence format in order to analyze our sequences later. So hang on to either the link or copy paste this into a Word document and keep this entire, including the whole sequence, keep that for later. And it's also important that you keep this little symbol here and the number. You can always search this number record for any of the sequences you have and go back to it at any point. It also gives you the little coded sequence and a little brief explanation of what it is you're looking at. So that's always helpful once you start compiling all of these different records. Trying to keep track of them is going to be important. Okay, so if you're feeling comfortable with what to do for this week with your gene, then you can stop this video now. If you would like a brief summary of what we just did, then you can stick around for the last little bit of this video. So we came to the gene database on the NCBI website. We searched for our gene of interest, and we selected the record that we're most interested in. Clicked on that record, and what we get is a full report. In this report, where we've been before to confirm that we have a protein coding gene, we will also come here to find out how many exons are in the gene. If there are multiple, we will look at the orientation in which the transcript is read and created. And then we'll come to this graphical view, select the green line in order to expand and see the purple and the red one. So the mRNA is the purple, the reddish one is the protein sequence. This week, we're focusing on the mRNA sequence. If you also have multi variants, then you'll see multiple green lines within this gene area of this graphical view. And if you do have multiple green lines, I want you to select the longest one and then select one additional variant as well. And we'll look into those records a little bit more. So what we did is we went to this mRNA record, we clicked on the graphical view, and it brought us to this page here. We save this page because we want to see the breakdown of the exons here. And then we also saved the Genbank record. That is this full record here. And we also saved the Fasta formatted record as well. All right, that's it for the NCBI demo. This should get you going for this week's project. If you have any questions, be sure to ask your lab instructors. You can always send them on the lab Q&A chat as well. All right, everybody, have fun looking up your gene of interest.